muscles last time, remember that? You want to know what the rotator cuff muscles are. Teres minor, infraspinatus, supraspinatus, and subscapularis. These four muscles make up the rotator cuff. As I said last time, a patient could have a tear in 1, 2, 3, or all four. When it's severe like that, it requires surgery. Now, I don't know if I mentioned this to you, but one way without doing any imaging is if you ask your patient if they're having shoulder pain and you ask them to reach in their back pocket as if to pull something out and it hurts, you can probably, you would, win a lot of money if you could bet on it, that they have a rotator cuff injury, okay, without doing any imaging. Introduction, okay, so muscular system is responsible for moving the framework of the body. In addition to movement, muscle tissue performs various other functions, and the muscular system performs other functions vital to maintaining a constant internal environment. So you want to know the three major functions of muscle. One is movement of the body as a whole, or movement of its parts. Heat production, and of course, posture. Okay, please know all three of those. So skeletal muscle contractions produce movement of the body as a whole or movement of its parts. Muscle cells, like all cells, produce heat by the process known as catabolism. What's catabolism? Breaking down, right? Breaking down. Catabolism and anabolism are two terms that's going to follow you into next semester. Okay, so I don't want you to forget what they mean. The heat produced just by one cell is inconsequential, but because skeletal muscle cells are both highly active and numerous, together they produce a major share of the total body heat. And again, skeletal muscle, it allows us to stand, sit, maintain a relatively stable position of the body while walking, running, or performing other movements. Next semester, I'm going to go into a lot more detail about how posture plays a big role in walking and running and all of that. So I'll go into much more detail then. Excitability, I want you to know another name for excitability is irritability, and it means the ability to be stimulated. The ability to be stimulated. I want you to know that contractility is the ability to contract or shorten. And that's easy to remember because if we make a fist, our muscles, our biceps are contracting, right? And it's shortening. So the ability to contract or shorten. Extensibility is the ability to extend. And when we put our arms straight out, that's what we're doing. We're extending or stretching. So we're going to increase the length. Because skeletal muscle cells are excitable, they respond to regulatory mechanisms such as nerve signals. So the term contraction, when applied to muscles, is meant in a broad sense of pulling the ends together. Muscles may extend while still exerting force. Okay. So we're going to talk about muscle contraction. And the first thing I want you to know about muscle contraction is that it has a lot of players, okay? A lot of players are involved for a muscle contraction to occur, okay? And you have to know them all. I think a good select all that apply question on the test is going to involve muscle contraction, okay? So you want to know that muscle cells are called fibers because of their thread-like shape. One of the first players in a muscle contraction is called the sarcolemma. The sarcolemma. And I want you to know that the sarcolemma is a plasma membrane of muscle fibers. I want you to know that definition. The sarcolemma is a plasma membrane of muscle fibers. The second player is the sarcoplasmic reticulum. The sarcoplasmic reticulum. One big function that I want you to know about the sarcoplasmic reticulum is that the membrane of this sarcoplasmic reticulum continually pumps calcium ions from the sarcoplasm and stores these ions in its sacs for later release. Okay, I want you to know that. Also in the sarcoplasmic reticulum, we're going to find the T tubules. That's another player. The T tubule, T actually stands for transverse. And these T tubules are found in the sacs, and their function is actually to allow the signal to go deeper into the cell. Okay, it allows the signal to go deeper into the cell. Now, I'm going to show you a visual in a minute. Here, you want to highlight calcium, because calcium is the major ion of a muscle contraction. I want you to know that. Okay, calcium is the major ion for a muscle contraction. Okay, for slide 9, the only thing I want you to focus on is satellite cells. And again, I'm going to give you more information about these satellite cells next semester. But for right now, if you can remember that satellite cells become active when we have an injury, or if we're weightlifting. So they become active when we're bodybuilding or weightlifting, and if we have an injury. That's all I want you to know about satellite cells for the moment.
When weightlifting or what?
muscle injury.
Thank you.
So let's talk a little bit more about these T tubules. You already know that the T stands for transverse. They extend across the sarcoplasm. And they allow electrical impulses traveling along the sarcolemma to move deeper into the cell. And it's very important for a muscle contraction. T2. These T tubules are actually sandwiched between sacs of the sarcoplasmic reticulum, and it's called a triad. T-R-I-A-D, triad. When the T tubules are sandwiched between the sacs of sarcoplasmic reticulum, they're called a triad. I'm going to come back to slide 12, but I want to focus on slide 13 right now. Okay, so here we have our sarcolemma, and that's a really nice visual. What are all these little guys here? These little mitochondria. And what do we know about mitochondria? Yes, the powerhouse of the cell, ATP. Remember, lots of ATP is needed for us to have a muscle contraction. Okay, so ATP, mitochondria, that's another player. Okay, gotta have ATP. Here's our sarcoplasm. Okay. And here in yellow is the sarcoplasmic reticulum. You see all that in yellow? That's all sarcoplasmic reticulum. And in between the sarcoplasmic reticulum, we have the T tubule, which is called the triad. And what's that T tubule do? It allows
Electrical impulses to travel along
good to go deeper into the cell. Okay, so it allows electrical impulses to go deeper into the cell. Then we have our sarcomere. We have our myofibril. This whole thing is a myofibril. And we have our myofilaments. Okay. You're going to find out in a minute that we have two myofilaments, and we'll talk about them in a second. So muscle fibers contain many mitochondria. Myofibrils are numerous fine fibers packed close together in sarcoplasm and run lengthwise along the muscle fibers. You also want to add that the sarcomere is the smallest contractile unit of a muscle fiber. So you want to know that as well. Each myofibril contains many sarcomeres. Okay. So on slide 15, I want you to know that we have two types of myofilaments. And they're called thick and thin. So we have a thick myofilament and we have a thin myofilament. I want you to know that thick is associated with myosin. Thick filaments are associated with myosin. Thin filaments are associated with actin. Okay, that's really, really important. Yep, thin actin, thick myosin. I want you to know that another name for skeletal muscle is striated muscle, and that's because it has striations to it. So sometimes we refer to skeletal muscle as striated muscle. Okay, we already said this, slide 17. We already talked about the triad. We already talked about striated muscle, bless you. Okay. So let's continue with myofilaments. What did I say about thick? It's associated with?
Myosin.
Myosin. Thin is?
Actin.
Actin. Okay. So in order for us to have a muscle contraction, there's 4 proteins that are involved. Okay? In order for us to have a muscle contraction, there are 4 proteins involved. And we're going to talk about all 4. The first one is myosin. Okay? Myosin is your first protein, and you already know that it makes up almost all of the thick filaments. And this protein has little heads, and I'll show you a picture. And these heads are known as cross bridges when attached to actin. Okay, so you want to know that myosin has these little heads that are known as cross bridges when attached to actin. Actin is your second protein. Okay, actin is your second protein. Myosin molecules are shaped like 2 golf clubs twisted together. Myosin heads are chemically attracted to actin molecules on the nearby thin filaments. So they angle towards the thin filaments. Okay. Are we okay so far? Which one has heads? Myosin. What are those heads called? Cross bridges. Okay, good. All right. So, we have 2 more proteins. We have troponin and tropomyosin. Okay, troponin and tropomyosin. Now, troponin is actually very important for you to know as nurses because of why? Heart attacks. Yeah, if you're a patient, if we suspect they had a heart attack, we're going to run blood work. And if that troponin is high, it's almost always indicative that they had some type of cardiac event. Okay, so troponin. Troponin is a cardiac enzyme, as well as being a prime player in muscle contraction. Now the 4th protein is tropomyosin. And tropomyosin. So now you know all 4 proteins, right? Myosin, actin, troponin, tropomyosin. Okay. So let's look at their function. First of all, here's the picture. Here's your myosin heads. Okay. So, and what they're showing here is that these little guys in blue here are the myosin heads. All of this is the thick myofilament. That's why I said, remember that the thick myofilament is attached. It makes up just about all of myosin, okay? So here's your thick myofilament, here's your myosin, and then these little pink circles represent the thin myofilaments. What's wrong? You sure? So here, they're going to, they're attracted, these myosin heads are attracted to the thin myofilaments. Okay? And I'm going to show you how this works in a minute, but are we okay so far?
What's the
Okay.
fourth protein?
Tropomyosin. So, thick myofilament, the myosin head. The myosin head is going to be attracted to these thin myofilaments, and it's going to pull it along. But I'll show you a picture in a second. Okay, I just want to get this down first. So actin is a globular protein that forms two fibrous strands twisted around each other to form the bulk of the thin filament. And if we go back to our picture, this is what we see here. That's the thin filament. Okay, the functions of tropomyosin and troponin. You want to know that tropomyosin is a protein that blocks the active sites on actin molecules. Tropomyosin is a protein that blocks the active sites on actin molecules. Troponin's job is to hold the tropomyosin in place. Okay? So troponin is going to hold the tropomyosin in place, and troponin is going to cover the active sites until we have a muscle contraction. Okay. So I want to go back to the picture for a second. Here's what I'm talking about. So here's your troponin, right here. Okay, remember, troponin's job is to keep tropomyosin in place. So this tropomyosin, which is this band here, do you see how it's covering the active sites? You see the holes there? Okay, so tropomyosin will stay on these active sites until we have a muscle contraction. Okay, and we'll see how that works in a second. But you have a little visual now, so troponin is right here, and its job is to keep tropomyosin in place. This is your tropomyosin, and this is your active sites. You see how it's covering them. Okay so far? Okay, all right. So what ion did I say is responsible for muscle contractions? Calcium. Good. Okay, you want to commit that to your memory. Okay. So a skeletal muscle fiber remains at rest until it's stimulated by a motor neuron. At the neuromuscular junction, which I'm going to show you a picture, motor neurons connect to the sarcolemma at the motor end plate. When this happens, a neurotransmitter is released. Have you ever heard of neurotransmitters? Yeah. What's a big one? What's one that you've heard of? Yeah, norepinephrine, excellent. What else?
Epinephrine?
Which one?
Epinephrine.
Yeah, epinephrine, serotonin, dopamine. Good, yeah, you got it, all right. Well, I'm gonna introduce you to the first one I think I talked about in here, and that's acetylcholine. Okay? So, in order for a muscle contraction to occur, the neurotransmitter, acetylcholine, is released into the synaptic cleft, and it will diffuse across the gap. It's going to stimulate the receptors, and it will initiate an impulse in the sarcolemma. We're going to talk a lot about acetylcholine next semester. You've heard of sympathetic and parasympathetic nervous system, fight or flight, and yeah. Well, acetylcholine plays a big role in that. Okay, but for right now, you want to know that acetylcholine is the neurotransmitter that is released when we have a muscle contraction. Okay. Here is an excellent visual of what I just talked about. Okay, so we come over here, we've got our sarcoplasm, we've got our sarcolemma. Now this is what I was talking about with the motor neuron. Do you see the motor neuron up here? Okay, so all of our neurotransmitters, all of them, they are in these little vesicles, okay? And the little purple dots represent the neurotransmitter. So in this case, the neurotransmitter is acetylcholine. So these vesicles are going to carry the neurotransmitter, or acetylcholine, to the synaptic cleft. This is the synaptic cleft. And do you see how the acetylcholine is in the synaptic cleft, and it's going to be picked up by these little receptors. Does everyone see that? Okay, so you got the motor and planar neuron, I should say the motor neuron. You've got your vesicles that's going to carry the acetylcholine into that synaptic cleft. They're going to attach to these receptors. Then we're going to have all this influx of calcium, right? Remember, calcium is the ion needed for a muscle contraction. Well, guess what that calcium is going to do? Because if we go back to our picture here, as long as that tropomyosin stays on the active sites, we're not going to have a muscle contraction, right? And what did we say troponin's job was? To keep tropomyosin in place, right? Well, when we have a muscle contraction and we have that influx of calcium, the calcium is going to bind to the troponin. And when it binds to the troponin, it's going to cause tropomyosin to go off of the active site so that we can have that muscle contraction. Does that make sense? Okay, so when you're studying this, I know it's a little difficult, but keep looking at the visuals, because the visuals are really going to help you. So let's go back, and here's what we see. We got the neurotransmitter, we've got that impulse going on, that's going to cause that influx of sodium. I'm sorry, not sodium, calcium. I don't know why I said sodium. Calcium. And once we have that influx of calcium, it's going to bind to the troponin, and it's going to cause tropomyosin to leave the active site. Okay. What do you think is going to make tropomyosin go back and to cover the active site? What do you think has to happen then? If calcium started it all, so what's going to happen We can't have a muscle contraction forever. We would be cramping all the time, right? So what has to happen for trypamyosin to go back covering the active site? What's gonna happen to the calcium? It's gonna go back to the sarcoplasmic reticulum, right? Because, and then everything will be relaxed again. That make sense? Okay. All right. So again, the impulse travels across the sarcolemma and through the T tubules where it triggers adjacent sacs of sarcoplasmic reticulum to release a flood of calcium ions into the sarcoplasm. That is what starts that whole muscle contraction. And then when the muscle relaxes again, then all those calcium ions are going to go back into the sarcoplasmic reticulum. So nerve impulses travels over the sarcolemma and inward along the T tubules. At this point, we're going to have a release of those calcium ions. The calcium binds to the troponin, which causes tropomyosin to shift and expose the active sites on actin. Once the active sites are exposed, energized myosin heads of the thick filaments bind to the actin molecules in the nearby thin filaments. So the myosin heads temporarily form cross bridges, you want to know that, between the thick and the thin filaments. After forming cross bridges, the myosin heads bend with great force, literally pulling the thin filaments past them. This is often called the power stroke of myosin. You want to highlight that, power stroke of myosin. Okay, so let's take it, look at it in steps. Okay, let's look at what's happening in this first picture. Where's the tropomyosin? Is it covering the active sites?
Yes.
It sure is, it sure is. So here's our actin. Okay, all these little, pink balls, red balls, they're actin. Okay. Here's our troponin. The tropomyosin is covering the active sites. Now, in order for this to happen, we have to have ATP, right? So we've got ATP. We're going to have ADP plus an inorganic phosphate. When we come over here, now what's happening? The calcium is binding to the troponin, and that's going to cause the tropomyosin to leave the active site. And now what we're left with is ADP and an inorganic phosphate. Here, what's happening here now, each myosin head then binds to an active site on the thin filament, and it displaces the remnants of ATP. And here's the power stroke I was talking about. So all that stored energy is going to cause this power stroke. And as long as that calcium is binding to the troponin, we're going to have a muscle contraction. And that's what we see here, that calcium is still attached to the troponin. We look at this under a microscope. Without calcium, that trypamyosin stays covering the active sites. With calcium, now it's moved from the active sites, and we're gonna have a muscle contraction. So when calcium is absent, the active myosin binding sites on actin are covered by trypamyosin. As soon as calcium becomes available, it's gonna bind to the troponin, and the trypamyosin is pulled out of its blocking position and reveals the active binding sites on actin. Are we okay? I know it's a lot, right? Okay. What neurotransmitter is involved in a muscle contraction? Acetylcholine. What ion is involved? Calcium. What's the job of troponin? Good, to make sure the trypamyosin stays in place. Okay. What's the job of trypamyosin? Yes, to cover the active sites. What happens when calcium binds to troponin?
The trypamyosin is active.
Good, yeah, it leaves the active sites, so we can have a muscle contraction. Okay. Is ATP involved?
Yes.
Is ADP involved?
Yes.
Inorganic phosphate?
Yes.
Yes, okay. They are all players for a muscle contraction. Okay, okay. All right. Okay. You need a break?
Yes.
Let's take about a 10-minute break and kind of absorb this a little bit. I think I'm gonna find you a visual.